All right, so it's 12 o'clock on my clock. And I saw Dr. Walter's already advancing his slides, so he's ready to go. So we'll go ahead and we'll get Grand Rounds started. Welcome everybody to Medical Grand Rounds, University of Colorado. Thanks for joining this afternoon. Just two reminders, no announcements from the department today, but two reminders. One is all Medical Grand Rounds are eligible for MOC and CME credit. So please use the link that Kelly Redard has placed in the chat to do that. And to please use the Q&A feature to ask Dr. Walter questions this afternoon. Myself and the chief medical residents will gather those questions and make sure that they get asked at the end of the talk as we have time. And now it's my pleasure to introduce Dr. Nick Walter. Dr. Walter is an associate professor of medicine in pulmonary and critical care medicine here at the University of Colorado, with a secondary appointment as an assistant professor in epidemiology, also here at Colorado. He holds an interesting educational and professional background. He's an undergraduate degree in economics and political science. He then obtained a master's in medical anthropology from Berkeley. He then got his MD degree from UCSF, where he also did his internship, residency, and chief medical residency. He then spent 2 years in the Epidemic Intelligence Service, the EIS, with the CDC and their respiratory diseases branch, where he did very interesting and important international work. And he arrived in Colorado in 2008 as a pulmonary and critical care fellow. And then as an instructor in medicine here, he also completed his PhD work in epidemiology under Dr. Tasha Fingerlin. He's an accomplished clinician at the Rocky Mountain VA Medical Center, as well as a recognized educator. His research is currently supported by a VA Merit Award studying the impact of sterilizing and non-sterilizing antibiotics on tuberculosis RNA synthesis. And he has been previously and continuously supported since his time as a fellow by awards locally from the CCTSI, as well as the NIH, the NIAID, the Bill and Melinda Gates Foundation, and the Doris Duke Foundation. So it truly is my pleasure to welcome Dr. Nick Walters this afternoon to speak about the progress and challenges en route to an improved tuberculosis treatment. Dr. Walter.
Great. Thank you, Jeff, and thank you for the opportunity. Today, I will talk globally about our current moment in tuberculosis treatment, and then shift to talk about how we in Colorado are contributing. I'm going to begin with a case of a 19-year-old man in Kampala, Uganda, who presented with cough. So this young man had had an episode of pulmonary TB 2 years previously. And as is the case in much of the world, that initial diagnosis was based on smear microscopy alone. He did not have the benefit of AFB culture or drug susceptibility testing. And he was treated empirically with the standard 2 months of isoniazid, rifampin, pyrazinamide, and ethambutol, followed by 4 months of isoniazid and rifampin in fixed dose combination. We do not have a record of his treatment outcome from his initial treatment. Currently, he was tested with the Cepheid Gene Expert MTB RIF assay, which detected the presence of M. tuberculosis, and additionally detected mutations that confer rifampin resistance. So rifampin resistance is typically associated with isoniazid resistance, and resistance to those two agents is the definition of multiple drug-resistant TB. So this is a process that begins in nature. There's a spontaneous, low-frequency random generation of mutations that can confer drug resistance. It's not particularly a problem until we select for it through inadequate treatment. So this is really an incremental, iterative process in which the bacterium acquires drug resistance mutations. Once resistance is acquired, we really kind of open Pandora's box, and then it can be transmitted. And that's really amplified by HIV, by inadequate infection control, and by diagnostic delays. If a patient is infected for the first time with a resistant strain, we call that primary resistant. So we don't know for sure, but I think it's quite unlikely that this young man would have acquired resistance during his initial therapy because he was on a fixed dose combination. So it's more likely that he has primary drug resistance. So how big of a problem is this? I'm going to review the pre-COVID baseline because during the COVID period, things have gotten so much worse that I think it can kind of give an inflated perspective. So each year, about 10 million people develop TB. And among those, about 500,000 are incident MDR TB cases. So the first challenge we have is a reporting gap. So if we think of those 10 million cases of TB, about 30% of them are never diagnosed and never reported. So those are occurring among people that don't have any access to healthcare or diagnostic services. So we know that they occur, but they're never counted, they're never diagnosed. So that case detection had been improving until the COVID pandemic. That had reached 30%, and now there's been backsliding, and it's now 43% are never reported as having TB. So then we have the burden that is reported. And among the people who are diagnosed with TB, we have a diagnostic gap. And I think our patient really represents that. So with his initial diagnosis was based on a smear. So we could tell that he had tuberculosis, but he didn't have any further testing. And of course, we can't tell from a smear whether that bacterium has drug resistance mutations. So about half of the time, we just simply don't recognize TB as being MDR TB. Then we have a treatment gap. So an important proportion of patients just simply never have the opportunity to start MDR treatment, even if they're diagnosed as having MDR TB. And as you'll see, our gentleman falls into that gap. And then finally, there's a success gap. So of the people who initiate MDR TB treatment, a little bit more than half are successfully treated. So when you add this up, of the 500,000 incident MDR TB cases each year, about 82% either receive no treatment or unsuccessful treatment. So we're in a situation where less than 1 in 5 of the new MDR TB cases that occur each year is cured. The remainder either succumb to their disease or continue to transmit the disease in the community. So as you can see, this is really presents a catastrophic snowball problem of drug resistance. So I want to situate our current moment in the really what we can consider a decades-long arms race between the bacterium and our therapeutics. A theme here is that really past is prologue, and the only constant is the development of drug resistance. So the first randomized controlled trial in history was a trial of streptomycin monotherapy. And that showed that the relative risk of death was reduced 75% within the initial months of starting therapy. Unfortunately, it also showed that by month 4, 85% of the treatment group had acquired streptomycin resistance. Streptomycin was quickly combined with para-aminosalicylic acid, and that showed that combination therapy could prevent the acquisition of resistance. And it really set up an axiom, which is that we never treat TB with a single drug, and we never add a single drug to a failing regimen. So in around 1950, isoniazid was introduced and added, and this became the triple therapy regimen that was the standard of care for about 15 years. That was an 18 to 24-month regimen. There ensued a period of vigorous clinical trial activity and innovation led by the Medical Research Council, which culminated in the development of what was called the modern short course chemotherapy regimen, which is the 4-drug standard regimen that we know today. So this has never been a good regimen. It's poorly tolerated, which causes problems with adherence. In practice, 6 months cures about 90% of patients, so about 10% either fail or relapse. It has an inherently low barrier to resistance. That is, if we have malabsorption issues or missed doses, it doesn't take that much to get resistant to. But nonetheless, that has remained the standard therapy for 40 years. So what ensued was a period really of complacency and neglect. And I think ultimately, in retrospect, it was hubris, because the sort of conventional wisdom with it was that the problem was essentially solved. This regimen was going to be good enough. It was thought that multiple drug-resistant TB was less fit for transmission, and so it might be problematic for the patient that had it, but it wasn't going to be a major social problem. We also have the problem of essentially divestment from the pharmaceutical community, who didn't see that TB had commercial. And so really this led to defunding of laboratory capacity and research, and there was never an effort to really globally roll out drug susceptibility testing. So I think it's quite clear now that a moment was missed. Thankfully, we are now in a period of renewed progress and innovation that I will tell you about after we return to this patient. So he has rifampin-resistant TB. We know that based on a screening test, which has identified that. But the problem is that there was no MDR-TB treatment at the time that I saw this patient, which is 2011. There subsequently is in Kampala. So there was no access to drug susceptibility testing. So we had no ability to identify, was he in fact resistant to isoniazid? Was he resistant to other second-line agents that might be available, like moxifloxacin? And so we found ourselves in this dilemma that tens of thousands of clinicians are in every year. Is like, do we risk adding, empirically adding effective drugs to failing regimens, and thereby potentially amplifying resistance for this individual and for the community? Or do we wait and wait for a more effective regimen? So he was registered on a wait list for an MDR-TB treatment program that didn't exist at that point and was discharged home on crowded public transportation and asked to stay masked. Unfortunately, I don't know his ultimate outcome. But I think it really, to me, his story, I still think about him, and it captures the dilemma that we face as clinicians and as policymakers. So thankfully, we are entering a new era of progress and innovation, which I certainly can't be comprehensive in summarizing, but I'll just highlight one major development that has been completed and a new, really provocative and ambitious idea. So in 2021, a 4-month regimen that substituted rifapentine for rifampin and moxifloxacin for ethambutol was non-inferior to the 6-month standard of care. So this was really a monumental, historic step forward in TB therapy. It really shattered the 6-month barrier that had been standing for some time. It's certainly a benefit for patients with drug-susceptible TB and for health systems that can shorten the duration of therapy. But it is ultimately incremental, right? We have substituted several drugs in the standard regimen, and it doesn't really fundamentally shift the balance of this arms race that we have with the pathogen. The moonshot idea is a pan-TB regimen, which would be a combination of 3 or 4 new drugs with new mechanisms of action that the bacterium has not been exposed to. So in concept, this would be capable of treating all forms of tuberculosis, whether currently classified as drug susceptible or drug resistant, because existing drug resistance really becomes irrelevant. So the goal is a new standard regimen that can cure TB in less than 4 months. So the NICS-TB trial demonstrated the power of this approach. So this was a trial among South Africans with extensively drug-resistant TB or treatment-intolerant multiple drug-resistant TB. So these are patients with advanced chronic pulmonary TB. Typically, they've been on antibiotic therapy for years, and they typically, in general, historically, have had a mortality rate of 80 to 90%. So the NICS trial treated these patients with 6 to 9 months of bedaquiline, which is a diaryl quinolone that inhibits bacterial energy production, protomidate, a nitronidazole that has some uncertainty about its mechanism, but probably is a cell wall inhibitor, and linezolid, a familiar oxazolidinone that is a protein synthesis inhibitor. And so remarkably, 6 months after completing treatment, 90% of these patients had a favorable outcome. So among the 10% that didn't, much of that was early mortality within the first weeks of therapy. So this is, for our field, this has been like a Lazarus treatment. It really is extraordinary. However, it's not a pan-TB regimen. It's unacceptable for general use because of its toxicities, neuropathy, and cytopenias primarily due to linezolid, and to its length. But it proves the concept that, if we apply new drugs in combination, essentially we can really shift beyond the current bacterial resistance. The good news is that the TB drug pipeline is now robust. This table provides a summary, and what we can see is that there's a number of candidate compounds and drugs, at various phases from lead optimization through phase 3. I'm not going to summarize all of them. Importantly, these are really very diverse mechanisms of action. And 3 new drugs, bedaquiline, delamanid, pratomanid, have FDA or EMA approval. The pipeline is moving. The other good news is that pharma is really now engaged, because if we think about a combination of 3 or 4 new drugs, it's almost certain that those are going to have to come from different pharmaceutical companies. And so what we need is these competing pharmaceutical companies to come to the table, to share information, to cooperate and collectively identify, optimal regimens, which remarkably, I think really even inspiringly, is actually happening. So there's a series of new nonprofit product development partnerships that bring together the major pharmaceutical companies, government, and private foundations like the Bill and Melinda Gates Foundations. Those are the Pan-TB Collaboration, the TB Drug Accelerator, ERA4TB, and Unite4TB. So these are all signs of progress. On the other hand, this is going to be far than straightforward. There's a very long road from individual new drugs to a new combination regimen that can become the global standard of care. And what I'll do momentarily is I'll really just at a high level outline some of the basic obstacles that we face. Among our challenges is that time is not on our side. If we take highly potent new agents and incrementally add them to existing agents, we are certainly going to generate drug resistance, and we risk squandering this opportunity. So if we think about how we could really shift the balance of this arms race with the pathogen, what we need is the element of surprise. We need to bring essentially a combination of new agents to bear simultaneously. So the first kind of bucket of challenges is the pharmacologic challenges. To be useful, a regimen needs to be oral and have once daily dosing. It needs to be tolerable. We need to avoid overlapping toxicities like QT prolongation or hepatotoxicity. And of course, our standards for toxicity have changed since the 1950s. It's unlikely that isoniazid would be approved today. We have to worry about interactions among the drugs within the combination, right? We may have one drug that induces the metabolism of another. We have to worry about the interactions between our drugs and other important drugs, and particularly HIV regimens. And then importantly, we need to think about whether drugs can concentrate in the difficult to penetrate sanctuary sites. And in particularly, we think about the caesium of the granuloma. So shown here is a granuloma from a C3 HEB-FHA mouse provided by the Robertson lab at CSU. And that caseous center of the granuloma, it is necrotic, it's avascular, and it's bounded by a fibrous capsule. So to access the bacteria that are residing there, a drug has to really diffuse some distance from the vasculature to concentrate in that area. That area is also lipid rich from the necrosis of foamy macrophages. And that, of course, is going to influence a drug's ability to essentially retain in that region. So this presents a considerable challenge, right? Because we have a couple challenges. If we do not achieve therapeutic exposure in the site where the bacterium are, we provide an opportunity for the bacterium to acquire drug resistance mutations. We can also easily imagine a situation where we get functional monotherapy. We give 4 drugs, but only one of them attains, it really penetrates to that region. The next set of challenges that is really our focus is the biological challenges. And a fundamental challenge is how we eliminate persisters. So when we treat tuberculosis, really the first thing our therapy does is debulks the disease. So when we treat with a standard regimen, the bacterial burden in sputum typically drops more than 99% within the first week of therapy. So the remaining months and months of treatment are required to eliminate a tiny residual subpopulation of bacterial persisters. That bacterial burden is often so low, as you'll see, that it becomes undetectable with standard measures, yet it is present and can return to cause treatment failure or relapse. So we need much better understanding of those persister populations and how they can be optimally targeted. Compounding the problem of persisters is that there's no single persister phenotype. This bacterium is exquisitely adapted to the conditions that it encounters, the diverse and heterogeneous microenvironmental conditions that it encounters during human infection. So we know that different compartments, or we'll strongly believe that different compartments have different phenotypes. And the phenotypic state of the bacterium absolutely affects the drug treatment. So we know that certain drugs, a given drug might be highly effective against a certain phenotype and have really limited effectiveness against another phenotype. So when we think about these compartments, we can think about it on multiple levels, right? So this is a bacterium that can affect any organ of the body, and each organ will have its own microenvironment. We can also think about lesional compartments within a single organ. And so if we really sort of dumb the histology down to, I think, to a cringeworthy level, we can just think about 4 basic lesion types in the lung. And generally, a given patient will have components of all of these. So they may have open cavities. They may have well-formed granulomas that are, for instance, that are interstitial, that are closed. They will have areas of pneumatic consolidation that is really a cellular infiltrate, and then they may have bacterium in the airway. And so those are going to be really quite different microenvironments on multiple levels. But one that is particularly important is access to oxygen, because this is an obligate aerobe. And so if we think of the open cavities of the airway, well, though the partial pressure of oxygen may be quite high, like 60. By contrast, that necrotic region of the inner granuloma is profoundly hypoxic. And this has always been thought to have a major impact on the phenotypes that adapt to those regions, and correspondingly an impact for treatment. Another level of thinking about compartments is whether the bacterium is intracellular or extracellular. Most of the time that it's intracellular, it will be intraphagosomal in macrophages, contained in that acidic and hypoxic compartment. We need to think about barriers to resistance, right? Optimally, a drug or combination, it would be resilient and robust, so that missing doses or having malabsorption would not easily lead to bacterial resistance. And finally, we really need to think deeply about, like, what does efficacy mean? What are we measuring and what is it telling us? So I'm going to shift to reflect on how our team here at the University of Colorado is contributing to this effort. So our campus is the coordinating center for a multi-institutional translational collaboration that we call the Consortium for Applied Microbial Metrics. And our goals are to advance molecular readouts that transform and accelerate how we select regimens. And in doing so, we are reshaping understanding of how antibiotics work and how to measure efficacy. So we are organized in a series of work streams, the first of which is in vitro, the bacterial physiology work stream led by Marty Boskiel in immunology and microbiology. Greg Robertson at Colorado State University leads the animal models work stream. I am Naheed at UCSF leads the human trials. Joshua Vasquez at UCSF leads immunology. I lead the molecular core. And then really this is all knit together through the knowledge integration work stream led by Radha Savik. And so what I'll present today is really the collective work of this team. So our team is thinking beyond burden to new measures of efficacy. So conventionally in tuberculosis, as in most other infectious diseases, we really thought about how the burden of the pathogen changes over time. And we can measure the burden based on culture-based approaches or molecular readouts. Either way, this is really kind of akin to thinking about the number of the enemy, right? If we were fighting a battle, we'd want to know partway through that battle, how are we doing? How much have we reduced the enemy and how much remains? We've taken a categorically different approach by focusing on how drugs affect cellular processes of the bacterium. So this is now phenotypic information. We're not measuring the burden. We're really measuring kind of the condition of the enemy. So by analogy, like, is the remaining enemy, are they robust, well-equipped, marching forward, or are they moribund and vulnerable? And if they are vulnerable, specifically, how are they vulnerable? And what we really want to know is we really want to know both pieces of information. These are really distinct but complementary streams of information. What we'll show today is that drugs do more than reduce bacterial burden. We can have two drugs or two regimens that induce the same change in bacterial burden, but result in different bacterial phenotypes and different relapse outcomes. Our goal is to accelerate development of drugs and regimens, working from the preclinical space all the way through phase 3 human trials. So today, I'm really just going to focus on some of our preclinical work. Our approach is to use the multiple new bacterial phenotyping methods that we've developed and then weave them together through a process of knowledge integration. I'm going to focus exclusively on one of those tools, which we call the RS ratio. So in Mycobacterium tuberculosis, the entire ribosomal operon, including the 16S, the 23S, and the 5S, are transcribed on a single transcript that includes unstable non-coding spacer sequence. The important aspect for our purposes is that that spacer sequence is rapidly degraded. So it serves as a marker of newly transcribed ribosomal RNA. So the RS ratio is simply the abundance of the unstable precursor, signaling new ribosomal RNA synthesis relative to the total structural ribosomal RNA. We multiply by 10 to the fourth to avoid leading digits. The high RS ratio indicates a bacterial population with a high degree of ongoing ribosomal RNA synthesis, and a low ratio indicates a quiescent bacterial population with low ribosomal RNA synthesis. So a fundamental principle of microbiology is that ribosomal RNA synthesis and bacterial replication are fundamentally linked. So the RS ratio really serves as a proxy for bacterial replication. So I won't show these results today, but we've shown this empirically in vitro and in animals. So culture, as measured by CFU and the RS ratio, really we're measuring different properties. And what I'd like to illustrate is the point that drugs do more than reduce bacterial burden. And so I'll illustrate that with results produced by the Bosco lab in vitro. So this is the traditional time kill curve in vitro. So on the x-axis is the days of drug exposure. On the y-axis is the CFU burden on a log scale. And what we can see here is that bedaquiline, which is a highly potent agent, doesn't seem to produce any killing within the first 4 days. And it takes a full 12 days to achieve a 2-log reduction. So if we were looking at CFU alone, the traditional measure, we would really get the impression that bedaquiline was having no immediate effect. When we look at the RS ratio, we get really an entirely different picture. So now I've added a right-hand y-axis and added the RS ratio. And so what we can see now is that inhibition of ATP synthesis by bedaquiline essentially immediately abrogates ongoing ribosomal RNA synthesis. So we can now measure really the molecular, physiologic, cellular effect of the drug long before we can measure its impact on killing. So this would be an example of a drug that doesn't initially change the number of soldiers, but has fundamentally transformed the physiologic state of those soldiers or bacteria. So isoniazid is really a counterexample. So here the x-axis extends only to 8 days because this is a potent bactericidal agent. We achieve a 2 log reduction in CFU after only 2.5 days. However, when we superimpose the RS ratio, we see that the change is really diminutive. So this is also on a log scale. So that's more than a 50% reduction, but it certainly isn't anything compared to what we see with bedaquiline. So this is an example of a drug in which the number of soldiers is massively reduced, but the surviving bacteria continue to synthesize ribosomal RNA. The physiologic state of the bacteria have really not been impacted to the same degree. We can see this in vivo as well, and I'll just give a single example from the Robertson lab at Colorado State University. So Greg's team infected Balb-C mice by a high-dose aerosol. They were then treated for 4 weeks with single drugs, shown here as isoniazid and rifampin. So in this plot, each dot represents a value from a single mouse, CFU value from a single mouse. And what we can see is that both isoniazid and rifampin achieved about a log reduction in CFU, and those were indistinguishable. We quantified the RS ratio in tissues from the same mice, and here we see that the phenotypes that result from that drug exposure are really very different. So they may have achieved the same decrement in burden, but they really are in a very different physiologic state. We have additionally used this to gain new perspective into complex drug interactions. And particularly, we have disaggregated the mixed TD regimen by looking at each drug individually and looking at the combinations of those drugs. So looking first at the conventional perspective, you can see that bedaquiline had a greater impact on the CFU burden than did pretomanid and linezolid. And the impact of pretomanid and linezolid on the CFU is essentially indistinguishable. When we look in the same mice at the same time points at the RS ratio, we now see that we can parse out differences that were not immediately apparent. We can see that pretomanid and linezolid are really affecting the bacterial population differently. Removing now linezolid and adding the combination of bedaquiline and pretomanid, what we see is that the combination of bedaquiline and pretomanid actually appears inferior and had less of a reduction in CFU than just giving bedaquiline alone. So based on the CFU burden, we would say that that's an antagonistic combination. So this is an observation that has been made over the course of a number of years in a number of different labs. It's been very consistent and been quite a puzzle, quite a paradox, because we know that this combination is highly efficacious in humans. It's really the backbone that is being considered for many of the pan-TB regimens. When we look at the RS ratio, we now get a different perspective on that interaction. We can see that the combination of bedaquiline and pretomanid actually did seem to have a greater impact on the RS ratio than either drug did alone. So we're getting different signals from different kinds of readouts, so different ways of measuring efficacy. What I won't have the time to show you today is that the RS ratio is associated with the treatment shortening activity of regimens. So that is to say that the regimens that are able to cure TB faster in mice suppress the RS ratio more profoundly and more rapidly than less potent regimens. These new tools provide important perspective as well at the end of treatment, particularly because they're more sensitive than solid culture. So these are results again from treatment with the mixed TB regimen. So there are groups of mice here treated for 4, 8, or 12 weeks with BPAL. And what we can see is with continuing treatment, we reach the limit of detection of culture. So everything that falls in that gray area is culture negative. And so in mice, as in humans, being culture negative does not mean cure. We know that there is a residual viable population there that is not culturable with standard techniques, because the cage mates that are given a 12-week drug holiday have the capacity to relapse. So we know the bacteria is there, we know that persister population is there, but it's just not measurable based on CFU, which is our conventional measure. And this presents quite a problem, because that is the critical point that is going to make the difference between durable cure and relapse. And we lose the signal at that critical point. Thankfully, we don't with the RS ratio. So when we measure the RS ratio in the same tissues, we see that we can really now detect a viable bacterial population through even the longest duration of treatment, and we can measure that treatment effect. So this for us has been like a shift from analog to digital in our capacity to really begin to parse that late stage of treatment that is so important in eliminating the persister population and achieving cure. So this also gives us new perspective at the time of relapse assessment and suggests alternative molecular outcomes. So in these models, relapse is assessed 12 weeks after completing treatment. So we will treat for a particular duration of treatment, then the mice are left without drug exposure for 12 weeks, then they're sacrificed, and we determine if they have microbiologic relapse or not. So what I'm going to show is results from two regimens. They're called the Simplicity TB and the Stand regimen. So BPAMZ is on the upper left, and PAMZ is on the lower left. So the difference there is just the presence or the absence of bedaquiline. We know that adding bedaquiline to that regimen makes it a highly efficacious regimen. And so what we can see is that in the upper left is that for mice that were treated for 4 weeks and then given a 12-week drug holiday, most of those mice relapsed. They were culture positive 12 weeks after completing treatment. If we give them another 2 weeks, so 6 weeks of treatment, most of them were now culture negative. And then if we treated them for 8 weeks, they were all culture negative at the time of relapse assessment. PAMZ is a less potent regimen. And so what we can see is that we need to treat longer to achieve the same outcome. So we need to, in order to really start to avoid relapse, we have to treat for longer than 12 weeks. And so this is the conventional measure. And based on this conventional measure that is used today to evaluate drug efficacy, we would say, okay, well, these two regimens, they achieve the same outcome. It's just a matter of time, right? PAMZ just requires a bit more time to get to the same outcome. We get a really different perspective when we use a molecular tool, a molecular measure of physiologic state like the RS ratio. This would suggest that we actually don't get to the same outcome. What we see here is in the upper right, in the same mice, we've measured the RS ratio. And for BPAMZ, we have not only sterilized those mice, they're culture negative, but we've also eliminated any residual molecular indication of viability. By contrast, for the mice that did not receive bedaquiline, at the lower right, we see that even though those mice were culture negative, they continue to have detection of the RS ratio, suggesting the presence of a bacterial population that continues to synthesize ribosomal RNA. So now we really have a different way of seeing efficacy. We can use these tools to probe drug action in lesions. And so as we talked about earlier, to be efficacious in a tissue like caseum, drugs have to first achieve a therapeutic concentration. And then once they've done that, they have to be active against the tissue-specific phenotypes that are present there. So each of these is necessary, but neither is sufficient. We need both. So we look at this in the C3 HEB FEJ mouse. which forms complex lesions that really resemble the lesions that we see in humans. And so that gives Greg's team the capacity to extract caesium from the center of these granulomas. We can compare that with the remaining lung, which is really a pneumotic cellular infiltrate. We can look at the spleen, in which the bacterium is intracellular within macrophages. And then we can take an airway sample via bronchoalveolar lavage. And as we had always suspected, but never really been able to show, we see that these key tissues harbor very different bacterial phenotypes. And so there's really a spectrum of RS ratio ranging from the airway, in which the RS ratio is quite high, consistent with rapid bacterial replication in an oxygenated environment, to the caesium, where the RS ratio is quite low, suggesting really a bacterial population that's replicating very slowly. So then the question is, is it the case that different drugs affect these phenotypes differently? So to get at that question, we looked at 3 drugs: rifampin, isoniazid, and bedaquiline. Rifampin is known to achieve a therapeutic concentration in caesium, and it is a potent sterilizing agent once it's there. Isoniazid is also known to achieve a therapeutic concentration in caesium, but it's a cell wall inhibitor. And we know that isoniazid really has limited activity against bacteria that are not synthesizing cell walls. Bedaquiline is a little bit different because we know that it has issues, pharmacokinetic issues, right? It is slow to load in caesium, but is sterilizing against slow-growing populations. And so when we look at a drug like rifampin, what we see is that rifampin decreased the RS ratio in caesium, and it decreased it in all organs. Right, so this is an example of a drug that penetrates, it accumulates, and it's active against the phenotypes that are there. That's our best case scenario. Isoniazid is a drug where we know it penetrates, but it doesn't seem to have any activity against the population that's specific to the caesium. It does have activity against the population of other tissues, but not caesium. So really, this illustrates the importance of really understanding those bacterial phenotypes. And then finally, bedaquiline is a drug that we know is slow to penetrate into the caesium, and lo and behold, if it does not achieve a reasonable concentration, then it doesn't seem to have an effect. It is potent in the tissues in which it achieves an adequate concentration. And so what we believe is that given more time, bedaquiline would have greater activity, but it just really, I think, illustrates the importance of the pharmacokinetic properties and the lesion decay in designing our regimen. So our vision here is to develop a lesional PK/PD model that will help us to understand how drugs and regimens penetrate, accumulate, and act. It's really that critical third component that we are adding. We are working closely with collaborators like Veronique Dartois that really have focused on identifying the PK component of this puzzle, and we bring the PD component. So the goal is to ultimately really identify the regimens that have maximal activity in these sanctuary sites. So these are new quantitative readouts, but they're really more than just a PD marker, because they're based on biological principles and really begin to suggest new paradigms for TB cure. So the traditional paradigm has been that the way that we cure TB is through sustained killing until we can gradually and incrementally eliminate the entire bacterial population. And so the way to shorten treatment is to speed the pace of killing persisters. Our results really kind of point to an additional paradigm, which is to say that, maybe inhibiting bacterial replication during treatment is an important component of achieving cure as well. Not only killing, but also stopping the input of new bacteria. So I'm going to close by reflecting once more on our patient to think through what has changed since 2011. So since 2011, we've seen FDA approval of bedaquiline, and there's increasing global access to both bedaquiline and to protonid and to linezolid. Kampala now has a strong MDR TB treatment program. And WHO recommends bedaquiline containing MDR treatment regimens. So that's all good news. But I think if I've communicated effectively through this presentation, I hope you'll really think to like what will come next. And what will come next, what is already coming, is the emergence of bedaquiline resistance. And so it turns out that the barrier to resistance to bedaquiline is probably not that high. And this is occurring in the context of monitored MDR treatment programs. And so this is really kind of a story that has no end now. I mean, the only way we can think of an end to this story is by making such a leap in therapy that we're able to introduce multiple new agents with new mechanisms of action that really get us ahead of the bacterium, or through something like the vaccine. So I'd like to really close by just emphasizing that this is an enormous collaborative team science effort led by the individuals at the top, but really depending on the contributions of these entire teams and our funders.
Dr. Walter, thank you very much for this talk. This is excellent. And we have lots of audience questions. They range from some sort of basic science of TB questions to some about your work, and also a lot of what you mentioned at the end, which is what's next? What is the future after, bedaquiline resistance becomes a highly emergent probability? One of the questions that relates to your science I thought was interesting is the audience is wondering about, whether this use of the RS ratio can be used for, or is being used for, other infections that have latent and undetectable populations and similar qualities to tuberculosis.
Yes, it's absolutely, in principle, it's not specific to tuberculosis. So Marty Voskuil is really leading an additional component that's focusing on non-TB mycobacteria, but we do see applications beyond that. It's really, our goal is really not to stop with the RS ratio. It's really to introduce kind of an era of molecular monitoring in which we don't think only about burden, but we really think about how compounds affect cellular processes of bacterium and really judge the efficacy based on that. And so that concept is not specific to tuberculosis.
And sort of a related question, having to do with a lot of the high science that's being talked about here that is able to be practiced at large academic institutions, but knowing that this is a disease, that obviously affects people in developing countries, many places which are fairly resource poor. How does this technology get deployed and developed in those countries and affect those populations most quickly?
Yeah, that's a great question. So our goal is to support the development of new regimens, right? Is to support the development of this pan-TB idea. So our initial focus, and I think that is a big focus, right? And that's where we want to keep our focus at the moment, so that we can help to create a new regimen that will be globally accessible to everyone. Beyond that, in terms of using on an individual patient level, there are a number of additional bridges that need to be crossed. Right? In order for it to be useful on an individual patient level, we really need to show that it's predictive for an individual patient, that we can test this patient and that will tell us this patient's outcome. And so that's just a much higher burden of proof. We aspire to get there someday, but that will be down the road to ask that question. So if and when we get to the point where we can say that this is predictive for this individual patient, then we can think about making it accessible. And the good news is that there has been a real rollout of molecular tools to under-resourced settings. And so the Cepheid Gene Expert is an example of that. So again, it was a private-public partnership that funded that, which detects tuberculosis and detects resistance, now detects more than rifampicin resistance. And that has been made globally accessible through concessionary pricing and things like that. So really the steps are first to show that it's useful on an individual patient basis. And then that would launch really a whole nother road of working to make it accessible.
A question that's come up as you were speaking, and you sort of mentioned the word at the end of your talk, and obviously vaccine science is in all of our minds right now. Can we keep people from getting this disease? The possibility of a vaccine, is that on the horizon?
Yes. I am really not the person to speak to this because it's not my area of expertise. It is, it would be the game changer, and there is an enormous amount of activity. There are challenges. Like one inherent challenge is that we know that natural infection does not confer immunity. So I could have tuberculosis today and be cured and be exposed to another strain or the same strain, years from now, and I might have another episode of tuberculosis. So natural infection does not confer immunity. There is progress, and but I probably won't be the one to provide a really cogent summary there.
Very fair. So let me ask you a question about the host. We talked a lot about the bacteria themselves, the mycobacteria themselves, and how they react. What do we know about the host's reaction with those agents? Why do some people reactivate, others will not? Those who reactivate, some become sick, some will not. How does that? What do we know about that?
Yeah, well, of course, we know that there are conditions which predispose to either infection or to progression from infection to disease. And those are the familiar ones that we think about, HIV, organ transplant, things like that. Then there are other conditions like smoking or diabetes that may confer a lower individual risk, but on a societal basis really are important drivers of the disease as well. Having said all that, most tuberculosis occurs among people who don't have any identifiable risk factor for progression to disease, right? And so most people who are exposed and infected with tuberculosis will not develop disease. They will proceed through their lifetime with latent disease and never have active disease. That subset that does progress to disease, there is some genetic predisposition, but it's relatively weak. So that question is not really easily answered. In terms of who relapses, well, there are important clinical predictors. So stage of disease is one. So burden of disease and how advanced your disease is to begin with. Then there are issues like absorption and what regimen and resistance and such. So it's a little bit of a mixed bag.
Great, thank you. And then one other question was asked about these various regimens, these 3 and 4 drug cocktails. And you talked about, some knocking the soldiers down, some knocking the soldiers out. And the question is about a different component, which is the temporal component, which is how do you add these medications? Is there any work being done on adding one at various times versus do they all start at the same time?
Yeah, that's a wonderful idea and a wonderful question. And it's an idea that has circulated for a long time. We think about, there's this initial phase that we call the bactericidal phase, which is this rapid killing at the very initiation of therapy. It's thought that that's really essentially wiping out the bacteria that are rapidly growing. And the drugs that work in that period really are different than the drugs that work against the persister population, right? So we think about isoniazid, it's a drug that really has the greatest efficacy against a rapidly growing bacterial population that's synthesizing cell walls. You wouldn't predict that it would have a lot of efficacy in the later phase of treatment when we think that the bacterial population's probably growing pretty slowly. So if we had the information, if we had molecular tools like ours that could really help us to understand these bacterial populations, then maybe we could progress to an era where we say, okay, for the first 2 weeks, we're going to concentrate on debulking the disease by giving agents that really kill rapidly growing bacteria, and then we're going to shift to a different therapy. And so really, our historical regimens have been developed really through, mix and match and really just seeing what works and what, we don't have 3 decades, right? That the MRC trials took 30 years to get to the short course therapy. And I think as you can see here, this needs to happen much faster. And so what we're hoping is that by providing essentially better information, we can drive this process forward to settle on a consensus therapy that will work better. But wonderful idea.
One other question about alternative agents is your lab or is anybody else looking at the use of, you talked about a lot of classical antibiotics that have different effects. Let's say things like monoclonal antibody treatments and non-classical ways to kill organisms within us.
Yeah, that has not been our focus to date. However, we do see the value of this type of phenotyping in understanding host-directed therapy and in host immune response. What our preliminary data shows is that when we compare models with and without effective immunity, essentially an immune milieu that is able to suppress replication, is able to control the bacterial population, well, we can measure it based on the phenotypic characteristics of that bacterial population. And so, one of the avenues that we aspire towards is really to be a PD marker for host-directed therapies or for immunity, for vaccines, for instance, because we would predict that a highly efficacious vaccine would enable immunity to better constrain and control bacterial replication. And we think we now have the capacity to measure that, but it's in its infancy.
Yeah. And one last question from one of your VA colleagues, and I want to throw it over to Dr. Chopra. Dr. Miller is asking, any progress in the area of immune modulation to improve cure rate?
Well, there is absolutely interest in the community in host-directed therapies. It's not my particular area of expertise, so I will not venture out onto that limb.
Fair enough. Sounds wise. And then Dr. Chopra, I just want to kick it over to you for any final questions for Dr. Walter.
Yeah, Nicholas, thanks for a wonderful talk. I'm going to sort of maybe zoom out just a little bit for you and ask you more of a broad question, which is sort of thinking about TB in an era of COVID. I've heard from so many people that, if we think about what has happened with COVID over the past 3 years in terms of our knowledge of the disease, our focus on therapeutics and where we are today, we've really come an incredible way in a short time period. And the comparison I've heard about is in TB, where that has not happened. And maybe, I don't want you to wax poetic on this necessarily, but give me a sense of what has the impact been on TB progress in terms of our focus on COVID? Have we lost ground in that respect? And is there something we can learn from our COVID response that you could take into sort of the world of TB going forward?
Well, COVID has certainly had a massive operational impact on TB care and caused heightened mortality and things like that. I think COVID has probably not affected the research domain. There's no evidence that funding has been taken away from TB to go to COVID. However, I really appreciate you asking that question because I think this is crucial, right? Tuberculosis is the #1 cause of death from infection in a non-COVID period, year after year after year after year, right? If we think about a disease like hepatitis C or HIV, transformative change in those fields, right? And as I think you can see here, we have potential to do it. The drugs are available, the process is moving too slowly. And it's a question of priorities, right? So in 2021, the funding for COVID was 140 times higher than the funding for tuberculosis research. This year, the funding for HIV research will be 5 times higher at the NIH than for TB, even though TB is the #1 cause of death from infection in HIV. So it really is a question of priorities. And if we can reorient our priorities to say that this is important and commit the resources, monumental things can happen, as I think the world demonstrated through COVID. It's just a matter of attention and intention to do that.
Well, Nick, thank you very much. It is 1 o'clock, so I want to be respectful of your time and the audience and say thank you again for a great presentation on an obviously incredibly important topic. Dr. Walter, thank you.
Thank you.